As of now, biological nitrogen fixation. Biological nitrogen fixation. It is the major source of nitrogen fixation, and it is done by prokaryotes, bacteria and cyanobacteria, either free living or symbiotic. Asymbiotic nitrogen fixation is done by the Azotobacteria and Klebsiella, and cyanobacteria like Anabaena nosta trichodesmium. Nitrogen fixation is done by symbiotic prokaryotes in the body of their plant host. Nasdaq, Anabaena, Anthoceras, etc. are symbiotic nitrogen fixing cyanobacteria, whereas Rhizobium frankia are symbiotic nitrogen fixing bacteria. Rhizobium in legumes and Frankia in non-leguminaceous plants symbiotically fix the nitrogen only under anaerobic condition by the root nodules. This requires cooperation of the node genes of legumes and nymph clusters of bacteria. Roots of legumes secrete the chemical attractants flavonoids and betaines. Bacteria collects over root hairs and release the node factors that causes the curling of the root around the bacteria. An infection thread enclosed in the bacteria is formed by root cells in response to the infection. When it reaches the deep into the cortex cells and it bursts, the rhizobia engulfed into the membrane enclosed symbiosomes within the cytoplasm. Synthesis of the oxygen from the cortical cells and cytokinin from the associated bacteria stimulates the nodule formation. Bacteria form irregular polyhedral structures. Bacteria inside the nodules. Legume host supplies the nitrogenase, nitrogen fixing enzyme which strongly inhibit the oxygen. Nitrogenase has more affinity to the oxygen than the nitrogen. Like the hemoglobin, an oxygen scavenger, is present in nodules which protect the nitrogenase from the oxygen. This is all about the nitrogen fixation. Thank you. Catch you later in next module.